Um, Hi everyone, my name is Liliana Jimenez, and I'm going to present an overview of my project, of my master project that's supervised by Dr. Kelly Brown from UBC. And it's titled Platelet Factor 4 as a potential marker for severe disease activity in GIA. So next, please. So GIA is the most common inflammatory rheumatic disease in childhood and describes a group of debilitating diseases characterized by chronic inflammation, pain, stiffness, swelling, and ultimately bone degradation. So there are 7 GIA categories, being polyarticular, oligoarticular, and systemic GIA, the most common subtypes and the focus of my study. Next, please. So the treatment for GIA, it's selected according to clinical manifestations and the disease activity of the patients. However, the severity of the symptoms can vary in each person, even if they are classified under the same GIA subtype. This makes even harder the decision for physicians to select an optimal treatment. And currently, disease activity can be measured using different tools to assess signs and symptoms related to inflammation. For example, the Juvenile Arthritis Disease Activity Score, also known as JADAS, includes for measurement 2 questionnaires answered for the physician or the patient, the count of active joints, and the measurement of non-specific inflammatory markers like ESR and CRP. Next, please. So in addition, several studies have suggested other proteins as potential markers to track disease activity. For example, the pro-inflammatory S100 proteins, which are mainly contained in neutrophils and are secreted after activation or by mitosis, which is a pro-inflammatory process where neutrophils release its own DNA along with other cytokines. Specifically, the heterodimer formed by S108 and 9 and the homodimer formed by S108-12 has shown to be sensitive measurements to track disease activity in GIA. This means that there's a high correlation between the circulating levels of these proteins and other measurements to evaluate disease activity like JADAS, for example. So other less studied cells in GIA are platelets and its proteins. And a study suggested that the mean platelet volume can be used as a marker to track disease activity in GIA because it was shown that the volume of the platelets are increased during the active stage of the GIA compared to inactive stage and healthy controls. And it is known that platelets with an increased volume can be considered pre-activated platelets and are more prone to release their content or to degranulation. So based on this, we focus our research in the proteins contained in the platelets, in the platelet granules, specifically CD62P and platelet factor 4. So as we most of us know, CD62P and P-selectin, it's commonly used as a surrogate marker for platelet degranulation, and it can be detected in the surface of the platelet by flow cytometry analysis. And PF4 is one of the most abundant proteins contained in the platelet and has been suggested to have a role in chronic inflammatory diseases by inducing the release of matrix melanoproteinase from juvenile chondrocytes. And these enzymes have been associated with collagen and tissue degradation in inflammatory arthritis. So also PF4 has shown chemotaxis effects in neutrophils and monocytes, which can also enhance the pro-inflammatory response. And studies show that PF4 can also induce mitosis, suggesting that there's a possible release of S100 proteins, as I mentioned in the beginning of the slide, after PF4 stimulation. So next, please. So as we know, platelets and its chemokines have a well-characterized role in hemostasis. Unfortunately, there's still a gap in the understanding of how platelets and the cytokines could be involved in GIA. And despite the urgent need for pediatric biomarkers, there are no validated means to identify children with GIA at risk of a more severe or progressive disease that might need a more aggressive treatment. So therefore, the aims of my project are to establish the first pediatric PF4 baseline in healthy children and compare the levels of PF4 to GIA patients and during the active and the inactive stage of GIA. and also evaluate a possible correlation between disease activity, the measurements of disease activity, with the levels of PF4 using JADAS. And finally, I'm also trying to evaluate a possible interaction between platelets and neutrophils. So we hypothesize that PF4 can activate neutrophils, and this might induce the release of S100 proteins that can further induce platelet degranulation and enhancing the pro-inflammatory response. So overall, these studies are going to be the first steps to assess the potential of PF4 as an early marker of a more progressive or severe JIA. And identifying these markers would allow clinicians to provide a more targeted and timely treatment for patients. So I think that's it. Thanks, everyone. Yeah.
Great. Thank you.
Yeah, that was my last slide.
Okay, that's great. Thank you, Liliana. Thanks very much. Okay, so we have time for some questions. Actually, I'll ask the first one. When you have really large platelets, the implication is that those are younger platelets. So is there an increased rate of platelet turnover in these patients?
Well, I haven't. The thing is that for JIA, there's not a lot of studies made in platelets. So I'm not sure if the age might be a factor there, but I'm sure they are. So there are more studies based on adults, not a lot of in children. So probably that's, we also hypothesize that platelets in JIA and in children are more reactive. So that's why we want to compare also that.
Okay, thanks. Other questions? Oh, I thought Dr. Overall would be all over the MMP question. All right, thank you very much. That was a very interesting presentation. We look forward to your data when they start rolling in.
Yeah, we're still recruiting, so yeah.
Yeah.